This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 334, recorded on April 24th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the only podcast all about viruses. Joining me today, that little laughter you hear, Dickson Despommier.
Hello, Vincent. You've changed the introduction slightly.
Why not?
That's exactly right.
You know any other podcast about viruses out there?
I don't.
Why not toot your horn, as they say?
Because otherwise no one else will, right?
You know, I'm looking at the weather here.
Yes.
It's freezing. I don't know why it got so cold.
You know what? It's a typical spring.
6 Celsius. And on my phone it says freeze watch for Putnam, New York.
That's true. That is true.
And you went fishing this week. Was it cold out there?
It was very cold. In fact, yesterday while I was fishing, it snowed. It was pretty cold.
Ridiculous. Also joining us from North Central Florida, Rich Condit.
Hey, how you doing?
That's a good change.
How's weather? Is it really nice down there?
Oh, God, it's just absolutely gorgeous. It's to die for. We got 85 degrees Fahrenheit, clear blue sky. That's 29.4 Celsius for those who might be interested. Clear blue skies, not really humid yet. It's early in the season. It is just a glorious day, and it has been for the past, several days. It's just great.
Fantastic.
Cool in the morning. We open up the house, hear the birdies sing.
Oh, shucks.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. When Rich came in, I thought he was the Fonz with that, hey.
That's it.
It's overcast here and 7 Celsius and breezy.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
Hey, Kathy.
You guys remember Chris Condit from ASM?
Mm-hmm. Yes.
Mm-hmm.
So he carpools to work, and his car partner, he tried to convince him to listen to TWiV. Maybe he's going to listen, and the guy doesn't want to listen to TWiV. He wants to listen to the Grateful Dead. So he made him listen for a while, and the guy said they should stick to the weather.
Hey, those guys are the Grateful Dead.
Do you know what the Deadhead said when he stopped smoking pot?
Uh-oh.
Man, this music sucks.
Yeah, right. Anyway,
so that was Chris's carpool.
Some people say we do too much weather and other stuff, but other people say we should just do that for the whole hour.
It's part of the rich tapestry of life.
You can please all of the people.
That's right. Say it, Rich.
Blah, blah, blah.
You got it.
This episode of TWiV is brought to you by SciMed Solutions, a leader in cutting-edge software that you would use in science and medicine, from EKG management. Has something to do with your heart, right, Dickson?
That's right. Electrocardiogram is what it stands for.
Why does it have a K then?
It's a good question, actually. Why does it have a K?
All right, let's do the ad, then we'll figure it out. From EKG management to sequence analysis, software built by SciMed helps researchers to increase their throughput. Do you have throughput, Dickson?
Only when I eat. We're not going there.
Throughput is stimulated by serotonin by orders of magnitude, allowing them to get results published more accurately and efficiently. SciMed has years of experience working with scientists in immunology, virology, and genetics. If you're going to this year's conference for the American Association of Immunologists, which is in NOLA, New Orleans, stop by and say hello in the exhibition hall, where you can learn about what SciMed can do for you. You can also check out their new product designed for life scientists. It's called the Dickson meter.
Tell them TWiV sent you.
That's right. You should say TWiV sent you.
You bet.
Yeah, because we're going to give them their website out, but there's no way they can track where the hits are coming from.
Tell them TWiV sent you.
Yes, that's a great idea.
All the TWiVes ought to go and tell them.
That's right.
TWiV sent me.
Of course, it is an immunology meeting.
Yeah, there's got to be
maybe autoimmunology will go. Is that the right way to say it?
Autoimmunity.
Oh,
Autoimmunity.
Whatever.
Autoimmunity.
SciMed software and consulting services will let you automate repetitive analysis and reporting of large data sets, streamline experiments, and maintain data integrity. You can contact them about research partnerships, database setup, mobile apps, and web applications. Go to SciMedSolutions.com. SciMed Solutions, innovative software empowering the world's leading minds. It certainly wouldn't be me, right, Dickson?
You don't have enough throughput.
I don't have enough throughput.
We can arrange something.
So here's the most amazing thing. I typed in EKG and Y, and the autofill choice was YK.
Wow.
Perfect.
So we are not the first to
So
ask
wait a
this
minute. Why
question.
does lysine have a K instead of a L?
L is
I
leucine.
know. Yeah, well, of course. And then lysine. I always
If
used
you
to
read
remember.
Dickson, you would know that.
I remember it being like-sine. I like-sine.
So, Kathy,
According to
Yes.
this Yahoo site, the K is from cardio in the German spelling.
Right.
did the Germans invent the EKG?
Oh, that's another search.
It does flow off the tongue better than ECG, right?
This is true.
Right. We have some follow-up. This is from Robin, who's following up about number 332, which was the first Vanderbilt episode. Thanks for the wide perspective. The co-option of host machinery to serve essential functions and the concomitant shrinking of genomes can follow two paths thereafter. If the invader earns its keep, it continues as an essential feature for the host, an endosymbiont or even closer, such as mitochondria, or the retroviral sequences for syncytin protein, both guaranteed transmission as long as there is a host with the syncytin sequence conserved due to its essential function. Where the invader confers no benefit on the individual host, it remains in a parasitic relationship. Even if such a virus is incorporated into the germ cell line, there may be no selection pressure to conserve the sequence. Perhaps all recognizable viral sequences in eukaryotic genomes are either recent or serve important, even if unknown functions, or both. On an ecological scale, even parasites serve the wider system, keeping things in balance. The biological imperative of every organism to increase its own biomass is at the root of our motivation to combat infectious disease. It remains to be seen whether Homo calidus can decide when enough is enough with regard to increasing its biomass and thus earn the title of Homo sapiens. As was mentioned, the further we stray from the ways of nature, the further we venture into the realms of the known unknowns, and even more ominously into the realms of the unknown unknowns, the roosting grounds of black swans. Wow, so
My
poetic.
Goodness.
right.
Well, not like that, but it's a little bit apocalyptic.
Just a little bit. All right, Robin, I do have one thing here. You say perhaps all recognizable sequences are either recent or serve important functions. So I don't know what you mean by recent, but we can recognize retroviruses in the human genome that we think went in 20 million years ago. It's still recognizable. It hasn't degenerated that much. So maybe over a few 100 million years, you might not be able to recognize it. But if an open reading frame is maintained, that we're going to get into that a little bit today with one of our papers. That might mean, for millions of years, that might mean that the reading frame is used like syncytin, for example. Anyway.
I think things can hang around for no particular reason for a long time.
So here's the thing. I just read the mutation rate. So when something goes in the genome, a viral sequence goes in the genome, you expect, I forgot the mutation rate. It's a very low mutation rate because actually when the genome goes in there, it slows down mutating because it's
Right. It's no longer a virus. It's
no
part of
longer
the
a
genome.
virus. Let's see if I can find it here very quickly. Well, I can't find it.
It would be the mutation rate for the third position in codons, roughly.
Yeah, something like that. Let me see if I hold on. Hey Dickson, you want to read the next one? I'm going to look for this. Okay, this one is for you, Dickson.
It certainly is. Sonny writes, Professor Dickson Despommier, this is in quotes, no, it's in capital letters, should be removed from TWiV.
All right, let me call the security here.
But this is actually not what they meant to say. It says, I am totally joking and actually would like to share my personal opinion about some comments that struck a nerve with me. I mean no disrespect for the CFS community and have no way near the required knowledge to comment on the disease, but I was taken aback by the listeners' letters by Claudia and Mary read on TWiV 331. I don't think Professor Despommier needs me standing up for him, but the disrespect towards him and other hosts really struck a nerve with me. This show is a public service in many ways, and to the best of knowledge, none of you are getting paid, and you are trying to inform the public to the best of your ability on a broad range of topics. Sure, mistakes are made sometimes. Dickson, my best Professor Racaniello voice.
That's Dickson!
Dickson! I can't even do myself right. That's how incompetent. No, that's not right. And most often corrected, Dickson. That is how science works after all. However, calling an emeritus professor of Columbia University uninformed, biased, insulting, and dismissive is in fact uninformed, biased, insulting, and dismissive in my opinion. Why do we need to personally attack the person rather than having a professional conversation and making sure the correct information is dispersed? As someone who wants to get into a medical field, I am aware that in fact self-diagnosis is something patients do and is indeed a factor in medicine and is not totally out of the realm of possibility since there is no specific case designation. A billing code is no proof of a definition, nor is one IOM report. I think the point that was trying to be made is that we don't know the exact mechanism or specifics, and more studies and more money is needed. But I do agree a clarification was needed and in fact was given. It is obvious something is going on and we just don't know what, and hopefully with time we can and help all of these people. I think the show has done a fantastic job in trying to inform the public on difficult scientific topics, and it is too bad that outside influences are not appreciative of the great effort that effort takes and further show disrespect at the host because they diverge from one's personal viewpoint or an agenda. I personally hope more research is done for ME/CFS and a better understanding can be had so people can be helped. Thank you, TWiV, for all you do. I thought it was important to let you know that the public service this show does is indeed appreciated by this 25-year-old male from Seattle who further appreciates science because of this show. Thank you. Best regards, Sonny.
Dickson, when I first read this, I saw the first line, and I said, I'm not going to read this. I didn't want you to feel badly. You know,
I would not have felt badly.
I wasn't going to read it if the tone was actually that you should be fired. Thank you, Sonny. All right, here's the mutation rate. It's a rate of 2 to 5 times 10 to the minus 9th substitutions per site per year, i.e. one difference between two LTRs every 200,000 to 450,000 years. So if a retrovirus goes in with two LTRs, you would expect to find one difference every 200,000 to 450,000. That's a long time.
It is.
So, you know, it takes a long time for it to totally lose everything. And, you know, one mutation may not do anything. It may take even millions of years to degenerate it. So, yeah, right, Rich, it can be there for a while. Alan, can you take the next one?
Sure. Robin writes, this is regarding ME/CFS. Maybe multiple different etiologies funneling through a narrow spectrum of symptoms, something like flaccid paralysis with different viruses or lumping all cancers together. The unwitting lumping of a heterogeneous group of conditions under one label could delay identifying the separate specific conditions. Maintaining alertness in this regard would be prudent. Yes, I think we've commented on that.
Yep.
There are, I would say, undoubtedly, there are multiple populations within that group that we can't distinguish at this point.
Yep. Rich, can you take the next one, please?
Who?
You, Rich Condit.
You, Rich.
Sorry,
the other Rich.
I've been here chatting away with my headset on mute.
Uh-oh.
You missed a lot of good conversation.
Oh, dear.
Mike writes, "Dear TWiVles?
TWiVles.
TWiVles?
TWiVles.
The trouble with
TWiVles wobble, but they don't fall down.
I thought he meant TWiVes and misspelled it.
Long-time listener, first-time caller. I am a mathematical epidemiologist at the Institute for Disease Modeling, IDM, in the lovely state of Washington, where it is currently overcast and 52 degrees Fahrenheit, 11 C. Yuck. I am one of the few people I mean, I can visualize this, right? We're talking, I wonder if that's Seattle. Sounds like it.
The yuck was editorial from Rich, by the way. It was not Mike's comment.
Right.
He might love it.
He might love it. I am one of the few people under the age of 50 who is paid to think a lot about polio.
Oh.
A group of us at IDM work with the Global Polio Eradication Initiative,
Sure.
GPEI,
Yep.
to provide analyses to support the polio eradication effort. On TWiV 331, Rich asked the question, are there clades of poliovirus, and amongst these clades, can you associate more neurovirulence among one or others? I remember asking that question. As Vincent suspected, it's easy and common to look at poliovirus clades, although people tend to say lineages or genetic clusters instead of clades. As part of the global polio surveillance program, every detected polio case gets its virus sequenced by labs with the Global Polio Laboratory Network. Clusters, i.e. clades, are used to keep track of importations, identify polio reservoir populations, and detect vaccine-derived polioviruses since the 1990s. Phylogenetic trees and sequences aren't routinely published, but there is roughly monthly sharing within the GPEI. A PubMed search for polio molecular epidemiology will get you some published examples. On to the question. The short answer is that there is no evidence for any differences in neurovirulence among circulating lineages of wild poliovirus within each polio serotype. From the pre-vaccine era, we know that there are differences in the neurovirulence, that is paralytic attack rate, across the three polio serotypes. From roughly 1 in 200 infections are paralytic for wild-type poliovirus type 1 to 1 in 2,000, that's a tenfold difference for type 2. We don't know why the serotypes have different neurovirulence rates. So there's that, but keep in mind that the three polio serotypes are better thought of as three different species rather than as variants within a species. Don't hit me. Within serotype, the only known genetic markers of neurovirulence are the attenuating sites in the Sabin vaccine strains. But most of the wild strains that have ever been seen have the neurovirulent nucleotides at all attenuating positions. The wild strains have lots of changes that are believed to be neutral, but none of those changes are associated with neurovirulence. By not associated, I mean that there is no epidemiologic evidence for differences in neurovirulence within the range of wild genotypes of a single serotype, and the non-attenuating changes that have been tested are neutral or nearly so on cell culture and mouse model phenotype assays. That having been said, there is a nearly complete lack of what epidemiologists call denominator data, and so the epidemiological case for no differences is really weak. That is, we don't know how many infections we're talking about. All we know is the number of cases. Essentially, all the sequences we have come from paralytic cases, so I don't see how we could tell whether some strains are producing more cases per infection or not. A low neurovirulence strain would just be detected at lower rates, and we'd never know. It's also worth pointing out that the low paralysis to infection rates for polio, let alone EVD68 or whatever is less likely, or likely less neurovirulent than polio, would make it really hard to detect differences in paralytic phenotype among strains. You'd probably need thousands of infections per strain, at least, to do the comparison. There is an interesting published example of polio doing something sneaky that was associated with a change in phenotype. Dexter et al., gives a link in Nature News, showed that an unusual antigenic variant of polio, wild polio type 1, that caused an unusually high case fatality paralysis to death rate was better at escaping prior immunity. I think we've discussed that before.
Yeah, we did.
I don't think partial immune escape is really relevant to the original question, but it's interesting. So that is a long answer to the question. Are there differences by lineage within polio serotypes? Maybe. I mean, probably yes, but not big differences. But I don't really have the data to know. Mike is a research scientist at the Institute for Disease Modeling. Thank you, Mike.
Yeah.
So a detailed answer to the question satisfies my curiosity. I appreciate that.
Does that institute get a lot of support from the Gates Foundation?
I do not.
Wondering.
Let's see about IDM. We'll see if they tell us. Blah, blah, blah. They don't say what their source of funds is. No. You would think so because they're up there, right?
Yeah, I would.
Same area.
I would.
Well,
Part of Intellectual Ventures Global Good Initiative.
Really?
Which that sounds like that's their umbrella. Intellectual Ventures is they look like maybe a venture capital firm.
They have malaria stuff, which is a Gates thing also. Maybe.
Yeah.
And they normally do investing in technology, looks like.
Yeah.
I'm sure Mike will let us know.
Just
All right.
curious.
Kathy, you are next.
On Wink, but first I have a follow-up. The inventor of the EKG machine was a Dutch person, Willem Eindhoven.
Okay.
Who probably does cardio with a K as well.
Well, I couldn't really find that using Google Translate. So I think it might be the German. So,
Okay.
whatever. Wink writes, poliovirus might have evolved to promote hind limb paralysis because this increases the chance that infectious stool will touch uninfected persons. Hadn't really thought about that before.
No, most of the transmission occurs from non-paralyzed people. Right. So the number that Mike just cited, 1 in 200 or 1 in 2,000, all those other people are the ones transmitting. So I don't think that would contribute very much.
Okay. 2, cancer on PBS was fine, but a bit of federal propaganda as well. It left the viewer with the impression that all was cured due to NCI research, never mentioning the Nobel Prize awarded to Gertrude Elion and George Hitchings.
Yes.
And we have a link to that Nobel Prize from 1988.
Yes, it was. It did not cover everything, that's for sure. I've heard a number of complaints about cancer. This is the Emperor of All Maladies show that I had picked. Well, I was excited because I liked the book, and I thought parts of the movie were okay.
Yeah.
All right.
It's going to be one of those things where if you're really close to it, you can see the flaws.
Of course.
You know.
All right, Gabe writes, I'm a longtime TWiV, TWIM, and TWIP and Urban Ag listener. Wow.
Gee whiz.
I especially enjoy how you insist that all of your guests recount their long, sometimes meandering paths into science. Anyway, I wanted to correct something you said in your TWiV 331 Passover Edition podcast. You stated that low CSF glucose levels is a sign of viral meningitis, when in reality, low CSF glucose is a sign of bacterial meningitis. My understanding is that the metabolic demand of viruses is not very high, whereas bacteria have more cellular mass to feed due to the order of magnitude size difference, so their metabolic demand is higher. In fact, CSF glucose levels are an important consideration in making a differential diagnosis between bacterial and viral meningitis. Then again, I'm a first-year medical student, so take what I say with a grain of salt.
He is probably most on top of the information compared to anyone else in the medical profession.
Yeah, I had gotten the CSF glucose from reading about bacterial meningitis, and I thought it would be similar, but it's not, so thank you. He's continuing. Another issue I found with TWiV 331 is that you glossed over the fact that EV68 is not found in CSF. I'm curious how EV68, or polio for that matter, exerts its neurological effects if it is not found in the CSF. Do virologists understand what is transmitting the pathogenicity of the virus from outside the blood-brain barrier to within it? Gabe, you don't transmit pathogenicity, okay? I know you're a first-year medical student, but probably your professors are teaching you that. Finally, in regard to your MECFS discussion, I wanted to reassure you that modern medical training puts a lot of emphasis on being compassionate and understanding with our patients. Love the podcast. Keep them coming, Gabe. Yeah, I would guess so, Gabe. It's really important that continue. However, as you know, or as you will find out, not every student who graduates medical school and is taught compassion actually learns it. So we do not have every physician being compassionate. Now, I found on Netscape the following. In rare cases, poliovirus may be isolated from the CSF or serum in contrast to paralytic illnesses caused by other enteroviruses. And I found a paper about the other enteroviruses. Conventional lab diagnosis of enterovirus meningitis relies on virus isolation from CSF or cell culture. This method is time-consuming and frequently unsuccessful because of the low titers of virus in CSF and difficulties in propagation. So there's not a lot of virus in the CSF. Nevertheless, I don't think it has to be there for the virus to be neurotropic, because we think these viruses get in via the blood, crossing the blood vessels into the brain proper, or through nerve transmission, so it doesn't have to be in the CSF, right? And it can go from cell to cell. So,
right. So it's in neurons, but it's not necessarily in the CSF,
right,
or
right.
It may even be in the CSF but not detected.
Right. I think it's very low, and I don't think it spreads through the CNS via the CSF.
Right.
Exactly.
That's the thing. Dickson, you want to take the next one?
Sure. This one is from Lance, who writes, Dear Family TWiVidae, I am partway through a very interesting TWiV 331. A couple of quick points. CFS glucose is measured to exclude bacterial infection.
CFS is chronic fatigue syndrome.
Yeah,
CSF.
CSF. This is, you could, this is very interesting because you have to read this extremely carefully. CSF glucose is measured to exclude bacterial infection. If the CSF glucose is more than 40% of the plasma glucose taken at the same time, bacterial infection, i.e. meningitis, is very unlikely, provided the person is not already on antibiotics or has only recently started. I'm not aware of any infections. You know, when you go fishing for 4 days, you forget how to read. And then these volume mute controls come on and it blurs out the rest of the words. Why am I getting that signal from my computer? I'm not aware of any infections where CSF glucose goes up. Not to say that they don't exist, but the main clinical use is to rule out bacterial infection, which is rapidly life-threatening and treatable. Concerning antivirals, after the onset of paralysis, I would not necessarily conclude that they are of no benefit without any data. There are many examples of treatment with antivirals after the onset of significant neurological signs and symptoms, which can lead to neurological improvement. For example, acyclovir for HSV encephalitis, and are clearly beneficial. I don't see why flaccid paralysis should be different unless the mechanism is immune-mediated and not direct viral damage to the anterior horn cell bodies of the spinal cord. There may well not be complete recovery, but given that neurons don't divide to replace themselves, anything you can give to prevent neuronal loss is likely to help. A lesser degree of disability is a 10-year-old who is going to carry that disability with them for maybe 60 or 70 years is worth giving treatment for. Many neurological infections don't recover as well as infections in other sites, part of the reason I think they're so interesting. But they can still be treated with sometimes remarkable recovery, but not always. Keep them coming, Lance.
All right. You're right. Although these entero infections are really quick, so I'm not sure how good the antivirals would be if we had some.
The window may be closed by the time we get there.
Yeah.
Alan, can you take...
But this is another good example of the physician's perspective of if you got a tool and it won't necessarily hurt and it has a chance of helping
that's right.
give it a rip.
That came up with the Tamiflu, remember?
Yes.
Yeah.
Yes. Yeah, and it's an interesting perspective.
All right, Alan, can you take that last one there?
Okay, Emery writes, Dear TWiV team, outside of D.C. it's 63 Fahrenheit, partly cloudy, and ridiculously full of tourists looking at cherry blossoms. that always, the cherry blossoms come out, and then the Cherry Blossom Festival is like a month later now, I think. So.
I'm doing the cherry blossoms next year because I'll be a free man and I can take off any time. So when they actually bloom, Ed will tell me. And I'll go up and do the cherry blossom.
Were they late this year, by the way, just out of curiosity? Because usually they've been earlier. Every year they've been earlier.
I was there last week. I was there Friday, and they were in full bloom.
Is that early for them, or is that late for them?
I don't know.
Do we know?
But I was at ASM, and they say, why don't you go look at the cherry blossoms? I said, no, thank you. don't want to look at cherry blossoms.
Jeez.
Why would I want to do that?
You'd rather look
That's
at plaques.
They're gorgeous.
It's like looking at, what are those flowers you like, Dickson?
Which ones? I like them all.
The ones you take pictures of.
I take a lot of pictures of different flowers.
The ones up in the Bronx.
You know those? Orchids.
Orchids. Why the hell do you need to look at orchids?
I get this all the time, kids. Help me.
What, do you
The
like to
cherry
look at-
blossoms are pretty, but I grew up in that area, so it's like, oh yeah, cherry blossoms.
Yeah, right.
The Cherry Blossom Festival this year was from March 20th till April 12th. So that gives you an indication when they thought the cherry blossoms would happen.
Exactly.
So they
So
were
they must
a
be
bit
late.
late. Yeah.
There's one in Brooklyn right now that's absolutely stunning.
I don't get it. I don't get what you want to
They're
look at.
pretty. They're pretty, Vincent. They're pretty. It's like
Such a curmudgeon.
I'm
It's
not
like
a curmudgeon.
pretty. It's like
Just some
pretty,
things
you
I
know,
don't
like,
get. Orchids and cherry blossoms, I don't get. Especially with tourists around. I don't want
it's a visual stimulation of your amygdala.
excuse the diversion. I'm sorry.
But that's
Yeah.
okay. My wife makes fun of me because I find daffodils annoying.
Really?
Oh,
How can you not like daffodils? They're so happy and they come up in the spring. Yeah, it's so annoying.
and they have a really nice fragrance.
Yeah.
Alan, you'll be
But-
pleased to learn then that they contain 2 poisons that I just read about in CNN News.
I knew it.
Chemical and Engineering News. It's got 2 of them.
You read
They're
it?
evil.
So don't eat them.
Dixon, you read it?
I actually read that. Yeah.
Wow.
Speaking of the Cherry Blossom Festival never quite hitting it right, the tulip time in Holland, Michigan also often doesn't hit it right.
Right. This was a long winter. That's why I
Holland,
asked.
Michigan.
Holland, Michigan. Yes, of course they would
true.
have a tulip.
That's right.
Under
So, Alan, you want to read the first one?
Yes, I could probably continue with Emery's letter.
Right.
In episode 331, there was a question posed about what kind of isolation precautions a child that presented in the emergency room would receive. Being a pediatric emergency room nurse at a level 1 trauma center, I thought I could share some insight, which is
nice.
exciting for me as my area of expertise is not generally helpful in the TWiV. This is why we love our listeners so much.
It's true. This is true.
The answer is a wonderfully non-specific, it depends.
Sounds like an Alan answer.
Masks are often available at the front door for those who wish to wear one or are encouraged by the staff, although compliance is personal and spotty, as you can imagine trying to keep a face mask on a 2-year-old to be a special sort of challenge.
Right.
However, the child you were talking about would be in a different category. Children with new-onset neurodeficits are not common. This or even moderate respiratory distress would draw the attention of the triage nurse immediately, as both conditions in a child require immediate evaluation. With the child roomed quickly at the attention of the combined medical staff, he or she would be removed from the general populace but not necessarily isolated. In the ER setting with a critically ill child, the emphasis would be on stabilization and very initial treatment, not achieving a specific diagnosis. With so many other common differential diagnoses, pneumonia, RSV, influenza, asthma, a novel pathogen is fairly low. House not being a great model for medical care. Standard precautions would be used, of course, but an unknown respiratory neurological illness would generally be on droplet precautions in a pediatric ICU inpatient environment. Droplet precautions, think flu, RSV, but not TB. Being pediatric emergency medicine specialists, I would say that we do a great job of identifying and isolating patients who present with even diffuse known infectious disease symptoms, measles, chickenpox, pertussis, and being so close to the capital, we have the luxury of having resources like negative pressure rooms and further training in infection control than a normal community hospital would. There should certainly be a higher degree of suspicion, and one of the greatest deficits in healthcare today is public health surveillance, preparation, and training. That being said, emergency rooms are a dirty environment, especially as we encourage those in need of medical care to just walk through the front door. And the little emoticon here that looks like it's a smiley face wearing a
It's wearing a mask.
Exactly. That's cute. That's very cute.
On the topic of medical research, I think one of the biggest differences is that in other scientific fields, saying "I don't know" is a recognized beginning of inquiry. In medicine, saying "I don't know," especially on the level of healthcare provider to patient, is often seen by the patient as a failure of that MD, NP, nurse not trying hard enough for both parties. Medical nursing practice and research is directly personal in a way that other sciences rarely are. Thanks for doing these papers. There were cases attributed to this outbreak in the area, and I enjoyed the team's discussion. Also, please let Dickson off the hook. I think his penance should be considered complete.
Thank you.
Until you're on the next hook, right, Dickson?
There's lots of hooks if you
there
fish.
There will be another hook.
There always is.
There's always a bigger hook.
A bigger hook. That's right.
All right. We have a bunch of stories here that accumulated in the last 2 weeks. So the first one was an article sent to me by my TWiV colleague, Michael Schmidt. So over on TWiV, we do these things called snippets, where someone will just spend 15 minutes talking about something.
We can't do that.
Yeah, I know. We can't do snippets. But he said
we would never snip.
Michael said maybe this is a snippet on the next TWiV. So it's a Wine Spectator article. I always wanted to cover a Wine Spectator. I don't actually read it, but I do like wine. I'd rather look at wine than cherry blossoms.
I've never understood the title of Wine Spectator, though.
Yeah, right. That's a funny name. Probably all the other good ones were used up.
Yeah.
Anyway, this is an article by Casey Carpenter. It says Pinot Noir's diversity may come from a 30-million-year-old battle with viruses. So I thought that was a cool title, and I got the research article, and we'll go over it a bit, but this is kind of a tenuous connection.
Yeah.
Yes.
Okay, I'm glad you agree with that.
Yeah, I think that
I thought
they must have
this was really overhyped.
I think the university sent out press releases. Maybe they sent it to Wine Spectator because one of the things worked on in the paper is the grape plant, right?
Yeah.
Right.
And they said this suggests this, and so they did an interview with the author, and basically what we're going to see is that there's some endogenous viral elements in these plants that have been there a long time, and they may have affected the plant in some way. So the title may come from a 30-million-year-old battle with viruses. Yeah, it might, but we don't really know. So it's very tenuous. Nevertheless, I thought it would be cool to talk a little bit about a plant virus.
Yeah,
sure.
So the paper is actually in Nature Communications, which I guess is their open access thing, right, Alan?
It is open access, I think.
Yes, I think that's right.
And the title is, it's published in November, actually. Endogenous florendoviruses are major components of plant genomes and hallmarks of virus evolution. The authors are gearing. What's the matter, Dickson?
Nothing. That's an interesting title.
Because that sound could either mean skepticism or interest. No. Gearing, Malmus, Copetti, Schwazny, Zwickel, Zintnicki, McTaggart, Scalabrin, Vezzuli, Wing, Cadenville, and Teixeini from Australia,
France,
Arizona, Guadeloupe, France, Italy. Yeah.
Anything from UC Davis there?
No,
I didn't see any UC Davis.
Okay, because one of the world's leading Pinot Noir
the Philippines,
grape
the International Rice Research Institute.
Oh yeah.
So basically, this is a paper with no wet experiments, which is fine. I'm just saying that. They just
in contrast to the fact that Pinot Noir is a wine, of course.
They look at many plants, not just grapes,
which
are Vitis vinifera. That's the grape, right?
Yep.
And they're looking at sequences. So there are lots of plant genomes being sequenced now, lots of them. So they decided to look through these and look for endogenous viral elements. And these can be retroviral, as we've talked about before, or almost every virus, a piece of almost every virus genome has been found in mammals. They're accidental integrations of just small fragments that allow you to date when they went in and when the virus was around, so they're very interesting. And in this paper, they're looking at a family of viruses. Not sure we have really ever talked about them. The Caulimoviridae, and one of the members is cauliflower mosaic virus.
Right.
They say they're the only family of double-stranded DNA viruses in the plant kingdom. Most of them are single-stranded DNA or RNA, right? Mm-hmm. These viruses actually encode a reverse transcriptase. They package DNA, but when they get in the cell, they make messenger RNAs, and then the mRNAs are packaged, and then the RT copies them into DNA before the virions are released from the cell.
Very like hepatitis B.
Yeah, right. So these are episomal. The DNA remains episomal. That is, it doesn't integrate. Yet, previously integrated pieces of these viruses have been found in plants. They're just pieces, right? They're mutated and rearranged. So they're not anything that's part of the virus life cycle, but they're there, and they can help you date when these viruses were around. And they may be beneficial, right? This is the big deal now about trying to find benefits of all these endogenous sequences. So they did a lot of sequence gazing, and they found lots of integrations that hadn't been found before. And one of the plants is the grape plant, and a lot of others. The rice plant they looked at.
Grape, rice, cotton, soybean, maize, peach, strawberry, potato, and tomato.
Black cottonwood.
So lots
of different plants.
What do all those plants have in common, do you think?
They're plants.
They're plants.
They got infected with this virus at some point.
More?
And they're angiosperms.
They're all
Thank
crops.
you.
What's
Kathy.
an angiosperm?
No, don't tell him.
What are you going to hold
Don't
back?
tell him.
Well, no. He obviously hates angiosperms.
As opposed to gymnosperms.
Exactly. Those are the ones that work out a little bit more.
Me. I knew
It's
this.
a flowering plant. It's a bloody cherry blossom.
See?
He doesn't know it.
Now you know why he doesn't like to look at the cherry blossoms.
It's the end of this paper. We're not doing it.
You might also, Vincent, know, and I know you know this, that the Pinot Noir grape is the most highly valued grape, except for the Champagne grape.
Why is it most highly valued?
Because they make wonderful, complex wine with it.
I tell you, I prefer
wonderful press releases that are very successul.
Did you see the movie Sideways?
No. I prefer Cabernet way over Pinots.
You do?
Yeah.
Why is that?
The Pinots are a little too thin for my taste.
Too thin.
So I went to dinner with Terry Dermody and his MD PhD students in Vanderbilt, and we drank Pinots all night, which I guess to satisfy a lot of people, you need to have Pinot, because not everyone likes the big red. I love the Tuscan reds,
You'd love this movie, though, Vincent. I might make that my pick of the week this week.
If I understand it correctly, the Vitis vinifera is all the wine grapes, and it's
Correct.
the cultivar Pinot Noir that we're talking about.
That's exactly right.
Yes.
So did they specifically look at the cultivar of Pinot grape here in this one?
They did.
Yes.
Anyway, they found a new genus of the Colymoviridae, which
That's exciting.
is called Florendovirus. All right, endo for endogenous, and Flora. Dickson, who is Flora? Yeah, not your aunt.
I think it just refers to Flora as a plant.
It's the Roman goddess of flowers.
Oh, really?
Flora.
No, I did not.
And
Vincent hates her.
I don't hate her.
That's right. Here's the thing.
No, I don't hate cherry blossoms. I just don't want to go see them.
You're going to get a lot of mail on this one,
It's
Vincent.
fine. You should see that there is a
A
mail.
lot of mail on this one.
We have an email which we'll read another time. Who is very unhappy that Alan and I like combustion engines.
That's true. That's true.
But we'll read that another time.
Okay.
And he's going to leave the show because of that. All right. So anyway, they found.
So does he prefer external combustion engines?
No,
or
electric. Electric. These cars like Dickson drive.
Oh, okay. So he wasn't too steamed about it.
All right. So they looked for the sequences, and they found lots. 19 of 25 in 2 out of 6 in the monocots, and 19 out of 25 in the eudicots, Dickson.
Yes.
And then they did lots of searching, and they basically formed this new lineage in the flowering plants, which they call florendoviruses. They actually were able to assemble a lot of complete genomes from these sequences. And again, they're not most of them are not integrated in one piece, although I think there's a couple of exceptions later on. So they have lots of figures where they can reconstruct the genome. These are interesting genomes. They have separate open reading frames. They also had evidence for a bipartite genome in 2 pieces, right? Because they could see 2 separate integration sites. They were able to do some dating.
That was nice for them.
Yeah, they did some dating.
It wasn't just that they were bipartite because of 2 integration sites, but that they had sequences that fit together that had some of the open reading frames on one and some of the open reading frames on the other. And in order to have a complete virus, you would have to have both of those chromosomes packaged in the so.
And they also were co-evolving too, suggesting they both went in.
Is this grape varietal polyploid?
I don't know.
They don't mention it, but it's really a minor focus on the grape.
Yeah.
So that would be like you would need to infect with both these particles to get a productive infection. Neither one would replicate on its own, but they would help each other. If that's all true.
Well,
since they're fossils, we don't know, but I had the impression that they're just both packaged.
Yeah, that's what I thought. They don't actually distinguish.
Yes, okay.
They don't distinguish, but I think...
Yeah, so this is an important point, is that as I understood it, there's no existing member of this family. These are all fossils.
That's right. That's right. Later on they say, we would have found these viruses if they were still circulating.
Right.
They say, we've been looking for plant viruses for 100 years.
So that's cool. Do we have any examples that we've gone over of this where we've discussed a fossil virus that is currently not in existence?
Well, the human endogenous retroviruses don't circulate anymore.
Yeah, okay, but at least there are retroviruses that we know.
Well,
this is a whole group.
But
right?
these are coliimoviruses. They're members of the Colimoviridae. So it's like Retroviridae, just a different genus. So I think that's okay. What's interesting, though, we'll get to the dating. So the rice colonization, at least 120,000 years ago, but some of these are even older.
Well, they can't go too far beyond that because flowering plants are only 130 million years old.
Yeah, well, they didn't even go that far. I'm talking about 20 million years or so.
Yes,
but an interesting question is, why are these old? Why aren't they around now? Right. So the same thing with the human endogenous retroviruses.
Well,
the last endogenization is about 1 million years ago. I mean, what happened? And I was talking to Steve Goff, and he said there are probably bursts of retroviral activity, and then it goes away. Maybe it lasts 100,000 years, it goes away, and then another few 100,000 years. That's very interesting. And maybe the same happens with plants.
Maybe. The other thing is to think about how they're transmitted, because
which ones, the plant viruses?
Yeah, because a lot of them use insect vectors.
Yeah.
Maybe their vector went away.
Maybe.
That's one of the things they speculate is that the particular
Yeah.
vector group was abundant but has disappeared.
Right. Because the rise of the beetles occurred prior to the rise of flowering plants.
Right.
And
The Beatles was like in the 60s.
That's true. That's exactly right.
And they have support for that because the Petunia virus, which is the nearest relative to these florendoviruses, has only been able to be transmitted by grafts.
There you go.
So V. vinifera, they say, is florendovirus rich.
My.
Has a lot of these insertions. 9% of the loci overlap with host genes, and 99% of these are within introns.
It caught it through the grapevine.
They heard it.
So this is I think this is one of the reasons for the Wine Spectator article, right? There are a lot of insertions in the grape, and being in introns, they say maybe it affects, gene function or activity in some way. They have no evidence for it, but it could, right?
At least they're in the neighborhood of places where you could have some effect on, proteins and by implication phenotype.
Right.
Wine makes a better press release, but it also it does so happen that in this paper that particular plant has an interesting thing going on.
So here's where I remembered they talk about what they call replication competency. Are any of these potentially infectious? So in the grape, they have different viruses. The A virus, the Vitis vinifera A virus, and C virus have decayed by mutation. So there's no loci containing an entire viral genome within a plant, or even a fragment with an uninterrupted open reading frame. So there's no way they would be infectious. But what is it? VVin B, I guess it's the B, is more likely to be competent, as many loci contain entire components and also uninterrupted reading frames. So I guess in a couple of instances they had the entire viral genome in one piece and an uninterrupted reading frame. So you could take it out and try and recover virus. It might be interesting.
Might be interesting to actually approach the two virus hypothesis by reconstructing those.
You could do that. So they say, Dickson, that the grapevine was domesticated around...
So would that be a gain of function?
I was just thinking that, you know.
No, but I'd be
if
sure
you
rooting for
resurrect
him.
some extinct virus, that could raise some hackles. here
Well,
and
you know,
there.
a HERV
People
K.
might start whining about it.
So the HERV K endogenous human retroviruses are the most recent ones to enter the human germline. They're about 200,000 years ago. They have complete open reading frames, and Paul B. Nash and some others have fixed the mutations that prevent infectious virus from being produced in humans, and they can recover this virus in cells, and they've been studying it. And there's absolutely no outrage about that. Although when you think about it, if this is something that was circulating 200,000 years ago, you know, we're not going to have immunity to it, and who knows, right? But they're working on it.
I think they did all that before the whole gain of function thing blew up, right?
Absolutely, absolutely. So Dickson, they say the grapevine was domesticated 5,000 to 6,000 years ago.
Oh, yeah.
So these intact insertions probably happened, you know, around then or more recently. So they're not that very old, which is why they're probably intact, right? What else is interesting here?
Did they give an origin for that domestication? As to geographic?
They just say the
origin?
Neolithic era. They don't say where.
Neolithic.
They do provide a reference, Dickson, if you're willing to read it. The paper is Biology of the Grapevine. Ooh, that's cool. It's actually a whole book.
How about that?
Would you like it for the weekend, Dickson?
I don't know. I'd rather drink it for the weekend than read it.
All right, so all of these florendos are in a lot of different kinds of plants, monocots and dicots. Discovery rate over 50%, so they're quite frequent. You know, it's interesting. We haven't picked these up until now.
Yeah.
I guess you have to look for it. 34 million years is what they think is the minimum age of this new Florendo virus.
34. That's
34 millions.
After the meteor hit.
Yeah?
Post-Jurassic.
So at 34 million years ago, Dickson, the Drake Passage opened up and permanent ice sheets formed in Antarctica.
Look at that.
I love it. I want to go
Look at
back and check it out.
And remember, there are no doctors
here's
waiting
what they
for
say.
you.
if these viruses still existed in an exogenous form, that is outside the genome and particles moving around, then they would have been discovered in more than a century of plant virology research, even if serendipitously as a contaminant. I don't know. What do you think? Maybe we could miss them?
Well, these guys don't think so.
These are pretty heavily studied species that they went after, though.
Yeah.
I suppose.
And I guess they're presuming that they would have some kind of effect.
Yeah.
You know, if there was a phenotype, then it would have been found out.
So they're wondering
Right.
now if these are beneficial. They say
Right.
maybe they help move genes around or they're sources of novel genetic material. And they say it's noteworthy that 9% are within plant genes, as we said, and these are within introns. So that's why they say overall they may have contributed to genomes and perhaps to the emergence of phenotypes that have been domesticated, such as in the grapes. And so that's where the Wine Spectator article comes from.
Right.
You know, all speculation, which is okay. I mean, I
they
don't think...
didn't look at barley, though.
No.
Well,
No, they
it's one of
looked
the
at
early
soybean
crops.
and cotton
Rice.
and Arabidopsis and
Yeah, but I mean,
rice.
there's this long-standing chauvinism in the literature in favor of wine over beer, and just
There must be a journal that's a beer spectator, right?
There's something about beer.
I know there are magazines about beer. I haven't read any. I don't know if it's
Yeah.
a beer spectator. Or,
They wouldn't call it spectator. They'd call
No.
it something frumpier than that.
yes.
But I'm not. I don't want to be harsh on the Wine Spectator. I think it's fine. They didn't really go too far from the truth, and they did talk to the investigators. So it's
They did
Besides,
actually, yes.
besides, it's bringing virology to the wine community.
Yeah, it's
That's
great.
good.
Here.
It's
Right.
Yes,
good.
and they did. I mean, it was clearly it stems directly from the way the press release was done, which focuses on the Pinot Noir grapes.
Yeah.
But there is original reporting in the Wine Spectator article. So
The other story I wanted to chat briefly about is, Ebola is still...
Wait, I just wanted to point out, actually I didn't time it, but I think we spent more than 15 minutes on the snippet. You'll have to tell me.
yes, I'm sure we did.
That was a
We're
complete
almost
sequence.
out of the show at this.
We're not going to read any more email, but we can get through these few stories. So Ebola has been lately in the news. There's been a lot of discussion about sexual transmission. And we talked a long time ago about how the virus can persist in semen, right? A couple of months.
Yeah.
And there's been some indication of sexual transmission previously. Now there's a new case. There's an article in the Times which is just from April 16th. Ebola researchers take new look at risk of sexual transmission. It's really a weird article, but first let me tell you what's going on here. There was a woman who, a 44-year-old food seller with no known risk factors who developed Ebola in Monrovia and died. Okay, and the country had had no infections for 3 weeks, so they looked at her boyfriend who had recovered from Ebola, and they found, I suppose by PCR, because they say genetic material in a semen sample he provided 175 days after he developed symptoms, 74 days longer than ever found before. So it says that they're trying to culture virus to see, and I like this. The Times, they write, they're trying to determine through culturing whether the man's sample contained virus, not just harmless fragments of genetic material. Yay!
Yes.
Good for them.
Unfortunately, the man refuses to give any more samples.
Yeah, he doesn't want to give samples, and this story goes through how they tried to get him to give samples, and you should read it. I don't want to discuss it here, but it's really weird, I must say.
So, as an interjection, if you went to the CDC website or the WHO website, would you get a clear picture of what's going on at this very moment?
You don't get a clear picture, Dickson, but they do say you better use condoms indefinitely.
I mean, the
Ebola.
Epidemic, has it since
we're not doing the epidemic right now.
No, I know we're not.
Doing something else. Just wait. Just wait.
But yes, the CDC does have comments on it, and they came out with a recommendation Sunday night.
Yeah, there's a rec, and they have a brochure which is extremely graphic about how to use condoms properly. And I've never seen anything like it on the CDC site. But it says, read this before you have... Ebola survivors, read this before you have sex. It's like a 2-page brochure. And basically the CDC says, we need to sort out what's going on here. So it's not just... The previous rec was 3 months, I think, after recovery. And now they say you better just use condoms indefinitely.
Wow.
But, the thing is, this is very rare, obviously. So in one of these Times articles, they quote a doc somewhere saying, what do you do with these outliers? Do you make general policy based on it? It's difficult. Anyway, what's this, Jens Kuhn?
Well, so there's a couple things that have been in the New York Times. To one article that then pointed me toward two different Nature articles, both from Tom Geisbert at UTMB. One is about results with the Vesiculovax vaccine in macaques for Ebola virus, and one about an siRNA treatment, also known as TKM Ebola Macona, and it's protective in rhesus monkeys as well. So that was one. And then in the Science Times just this last Tuesday, a long story discussing the evidence that Ebola was potentially in that region for a long time. There's some sero evidence, but they do point out and interview people who say, well, that's sero evidence, and that might be some kind of cross-reactivity. But also some genome phylogeny evidence that I think we already knew about, but is kind of restated here. And then, there's a picture of Jens Kuhn and,
Peter Pio. So, Jens, when we talked to Jens on TWiV, we mentioned this serological cross-reactivity that had been picked up elsewhere, and he kind of was very skeptical, if you remember, at the time, and I guess also here,
But again, if you start putting things together, both the serological data and the phylogenetic data, it maybe points to something.
Rich, is there a sit rep here?
Kathy actually dug it up, but it's the decline in confirmed cases of Ebola virus has halted over the last 3 weeks. So in the week of April 19th, there have been 33 confirmed cases, compared with 37 and 30 in preceding weeks. That's none in Liberia, 21 in Guinea, and 12 in Sierra Leone. So it's not over.
Yeah, it's not over.
And I've thought about this some. years ago, if there were 33 cases of Ebola in a given week, everybody would freak out.
That's true.
Yes.
That's true.
That's right.
And now there's no news at all. Yeah.
Yeah.
A little
Well,
bit,
our expectations have been totally recalibrated in this outbreak.
Right.
It's probably why I asked my question.
So the point they make in the sit rep is, in many words, is simply that these last few are tough to squash. It's not over.
So some of the phase 1 trials of vaccines have been completed. They've been done. That's been done here in the U.S. But do you think it would be justified to go in there and immunize a few hundred people?
I would think so. Yeah.
I don't know if they're.
The problem
to
is
that.
you're not going to probably get any efficacy data out of that, right?
Right.
You're gonna get safety data, and you're probably going to get data. You'll bleed them and find out whether they make antibodies.
But you might as well do
depending
those
on the
studies-
vaccine.
You might as well do those studies someplace that you can control better and it's more convenient. So I think at this point there are still cases, and that's a problem. But they're going to be so scattered that if you went in and you vaccinated a bunch of people and you still saw cases, it wouldn't tell you anything.
Yeah.
Well, but you could think about a ring vaccination strategy.
Right. That's what I was
with
thinking.
the contacts. If you could do good enough contact tracing,
Yeah.
and then maybe do the ring vaccination with good informed consent.
Yeah.
That's what I was thinking about, yeah. And so I'm wondering if people are thinking about that because this is continuing and they want to be able to test the vaccine, so they could do that, yeah. All right. Next story was sent to us by Anthony. He sent us an article about a outbreak of canine influenza in the Chicago area. It's now spread to Wisconsin and Indiana. So this is a pretty big outbreak of over 1,000 dogs. So right away I went to my source, which is ProMed Mail, to find out the real thing. So this is an Asian strain of canine influenza H3N2 not seen before in the U.S. Over 1,300 dogs in the Chicago area and spreading from there. The ProMed Mail article is interesting because they try and talk about how the virus might have arrived in the U.S. because it's not been seen here before. And they said there's some rumors that some dogs have been rescued in Asia and brought here, and that might have imported the virus, but they say there's no evidence for that. And they suspect that, or they propose that sniffing dogs, which are at airports, might have done it. So they say a sniffer at an Asian airport might have sneezed on cargo. The cargo gets flown to Chicago, and then another dog sniffs it and picks up the virus and then spreads it. So that's interesting. And this is how these infections these infections typically get spread in kennels where dogs are close together, right? There's a really good CDC article about this that talks about it. These are, again, 2 strains. They don't infect people as far as we know. A long time ago, we talked about a dog, greyhound influenza virus outbreak that was occurring.
Right.
And that came from horses. So one of these canine influenza strains, H3N8, not the H3N2, which came from overseas. The H3N8 originated in horses and then spread to dogs. And we did a story on TWiV about it going from horses into greyhounds. I don't know if anybody remembers that. It was a long time ago.
Yeah.
So this is not a really serious infection in dogs. They have symptoms. Many of them
they get a cold.
They get a cold. Some of them may get more serious than others. It's spread by, pretty close contact. It's an aerosol, but coughing and sneezing. And it's typically, if you walk your dog and you don't see any other dogs, I don't think you can get it, right? Because there's no virus around.
Right. There's some probably asymptomatic infections, very few deaths. Right. So it's not that big a deal. Your dog just gets sick.
Yeah. So if your dog is coughing, you could bring it to the vet and they could do a test. There is a vaccine, actually, for the U.S. strain, not for the other one that just arrived. So it's interesting the way they think it might have arrived, though. I think that's sniffing dogs.
So the sniffing dogs theory and the other, which got some press because it's a little bit more sensational, I guess, was the rescue dog theory, and apparently there are organizations that, or at least there's one organization that goes to Asian meat markets and buys dogs that are being sold for meat, or somehow rescues them from there, and then they transported some number of those to the U.S., but that doesn't appear to, at least at this point, that doesn't appear to account for the distribution of.
Right.
Yeah, most of those dogs went to places where the disease is not.
Right. Right. So it's not like they all came into Chicago, or any of them came into Chicago, but you do see this outbreak in Chicago, and so it would seem to make a little more sense that it was something air transport. I don't know. I mean, you'd really only need one introduction.
Yeah, sure. That's all you need.
So there's also the much simpler possibility that this was just somebody's pet being imported.
Yeah, they say there are pretty strict requirements for getting a pet into the U.S., right?
Right.
Of course, if it were asymptomatic, then that could happen.
Yeah,
right?
you bring an asymptomatic dog in, and I know there's some quarantine period, but it's going to be quarantined at a kennel?
Right.
Yeah.
So maybe that's not completely airtight.
So the incubation period is 2 to 3 days.
Right.
So that should show up in a quarantine, which is probably longer than that.
Okay.
So evidently this,
if it's symptomatic in all cases.
this one this new outbreak is the H3N2.
Right.
Is that right?
That's
Okay.
correct. Yeah, that's
Because there's already a vaccine for the H3N8,
Yeah.
but they don't know whether that
Right.
cross-reacts.
Yes, the H3N2, there's no vaccine, and that's the one that's been brought in that had never been here before, right?
Because otherwise I was going to say, make sure all the sniffer dogs are vaccinated.
Yep. All right, so that's kind of cool. Now, a story here that has been locally a big deal, right, Dickson, is
Indeed.
has to do with one of our colleagues here.
It's true.
I just thought I would mention it because he is a Columbia University faculty, Dr. Mehmet Oz. As
Yes.
everyone knows, he's got this TV show where he
Promotes quack. I'll say it.
promotes quack treatments, right?
Yeah.
He is an MD. He's a surgeon, and he got
A
promoted.
surgeon of significant repute.
Yes, he's
Yes.
very
Yeah,
good. My
yes.
colleague here, Saul, said he was a fantastic
I actually know
cardiac
him.
surgeon.
I've talked with him. And,
Really?
in fact, once he invited me to go on Oprah with him to talk about parasites, because that's what he was going to talk about. So I just supplied the parasites for him, and he went on by himself.
Anyway,
Yeah,
so
but there's much better money in hawking snake oil than there is in cutting fences.
so
This is true.
A number of physicians from all over the country wrote a letter to our dean, Lee Goldman, and they say, we are surprised and dismayed that Columbia University College of Physicians and Surgeons would permit Dr. Oz to occupy a faculty appointment, let alone a senior administrative position in the Department of Surgery. He's guilty of either outrageous conflicts of interest or flawed judgments about what constitutes appropriate medical treatments, and they want him to be fired. And, of course, Columbia said, our director of communications here said, he's in a university and he's allowed to say what he wants. That's the protection that we afford.
He earned tenure before he went into the business of hawking quack cures.
Yes,
is the
he's
upshot.
tenured. He's tenured, so he can't be
He's
fired
tenured.
for
They
being
can't get
stupid.
rid of him. So there he is.
And, whether you think he is hurting Columbia's reputation or not, it may not look good for us, but I don't know that anything will change unless you can prove that he's harming people, right? And that's hard to do, I think.
Right, and he did legitimately earn tenure at Columbia. I mean, as
he did.
said,
Yeah.
he's a great surgeon, and would have been nice if he'd stuck with that as his main line of work. But so this is the downside. One of the downsides of tenure is that you can tenure people, and then they can go off and say stupid things that make the university look bad. But
we've complained about him before, and I do think he's a problem. I never thought he was a problem for the university because of what we just said. I don't know how much work he actually still does here. Do you know, Dickson? Does he still do surgery?
I do not know. I know he's still on staff, so I think he has to practice at least 2 months out of the year in order to maintain his presence.
Now, he did, Oz questioned the credibility of these individuals. I don't think that's really a smart thing to do.
I don't think he's in a position to question.
Yeah, really? Anyway.
Right. He evidently spent, from one thing I read, half of his 1 hour show discussing this. So at least he had to spend time on that where he couldn't be spending it on some other things.
It's called damage control.
Yeah.
I'm asking myself why people get weird like this, but I guess it's money and fame,
money
right?
and fame. Yeah, he says this stuff, and it keeps him famous, and that's going to be powerfully addictive.
Wow.
So I found one BMJ article that did an analysis of TV medical talk shows, what they recommend, and the evidence to support their recommendations. And just tell you the conclusions. Recommendations made on medical talk shows often lack adequate information on specific benefits or the magnitude of the effects of the benefits. Approximately half of the recommendations either have no evidence or are contradicted by the best evidence available. Potential conflicts of interest are rarely addressed. The public should be skeptical about recommendations made on medical talk shows. And
Yes,
they
the public
analyzed a certain number of 40 episodes of Dr. Oz and 40 episodes of The Doctors. Show I never heard of. So
God, what an awful project to have to do.
So our communications officer wrote, as I am sure, to these physicians who wanted him fired, as I'm sure you understand and appreciate, Columbia is committed to the principle of academic freedom and to upholding faculty members' freedom of expression for statements they make in public discussion. And so that's why we're allowed to do TWiV, Dickson.
Here.
But we don't give medical advice.
No.
We
Try not to.
We don't.
I mean, and the perspective one could take on this would be, don't hate the player, hate the game. Mehmet Oz is in that position because there's a demand for people to listen to reassuring but more or less BS solutions to their problems, and somebody's going to be doing that.
Yep.
If there weren't drug users, there wouldn't be drug dealers.
There wouldn't be drug dealers, exactly. So he's selling that, and he's the one who happens to be doing it. But if he weren't, well, in fact, there are others who already are. So it's not a unique case.
All right, the last thing I want to mention is an op-ed that was published in the New York Times by Newt Gingrich, who's called for a doubling of the NIH budget. He makes the argument that since the budget was doubled, some time ago, the budget's been flat, which has been resulting in really a 20% decrease because of inflation. And this is bad because science is really important for our health and so forth. He echoes all the things we often say here on TWiV. We are in a time of unimaginable scientific and technological progress. By funding basic medical research, Congress can transform our fiscal health and our personal health, too. I've never agreed with Newt Gingrich before, but I have to say I agree with this.
What's Newt doing nowadays?
I don't know what he's doing.
I think he's still a history professor, and he does stuff like this. He's a pundit. I interviewed him a few years ago for a story on science education, and he is actually he hasn't always been pro-science in all of his rhetoric because he's also a politician, but he's actually a lot better than most of his ilk at supporting science, and he was very much in favor of the original NIH doubling, and. When we were talking about science education, he's very pro-science education and thinks that creationism needs to be taken out of the science curriculum and that sort of thing. And so he has a lot of positions that people who don't align with him might not expect.
Well, good for you, Newt. Thanks.
Yeah.
Yes,
This is good.
The only thing I would say when I think about this, my fear. When the NIH budget was doubled, they kind of blew it. Okay, they being the consumers, because the universities built a bunch of new infrastructure, hired a bunch of people, and then the doubling, leveled off, and now they got all this extra activity and no sustained support. So I think that hopefully I can say when the NIH budget increase happens, I would like to see some sort of more sustained increase, and that it be somehow moderated or regulated in a fashion that the money gets spent more appropriately. I think stick it all into RO1s,
Yeah.
Yeah, so someone over on Facebook on the ASV page said, if this goes to building university buildings, it wouldn't be a good idea. And
exactly.
It can't happen. It's got to be for research, absolutely.
Right, and if it also if it just goes to expanding the population of graduate students and postdocs who, when it gets flatlined a few years later, won't be able to get jobs, that's not going to be helpful.
No.
Well, that's the important thing, is that it doesn't get flatlined a few years later.
Well,
There's got to be some sort of long-term strategy.
Exactly. There are the two components here. One is there has to be a long-term strategy for at least maintaining it and adjusting it for inflation, and two, you have to get the universities to back off from the trough a little bit and moderate their approaches. But
that can probably be controlled by how the money is budgeted
that
out
can
of
probably be
NIH.
regulated. Yes.
All right.
Thanks, Newt. Thanks again. Keep them coming.
Our other sponsor for this episode is the ASM Education Department. We want you to know about a special section of the Journal of Microbiology and Biology Education having to do with scientific ethics. This is how many articles? 45 articles section, which gives you a look at ethics in science, including the responsible conduct of research, ethics in science education, publishing, and authorship, and ways to incorporate ethics into STEM education. And these articles give you ethics-related activities and tools you can use in a class or in a laboratory. This is essential, this stuff, and you have to know why, and they give you the articles that tell you the potential influences, promises of funding, research competition, authorship that can compromise research integrity, articles on why responsible conduct of research training is necessary, and how to incorporate and evaluate this into your daily life. So if you want to see this section, you can go to jmbe.asm.org.
And then you might have to search a little bit, and I'm trying to find the link that I found and sent to training grant people here. But it's an issue that was published, I think, sometime late in 2014.
Yeah, you have to, because you can't get a direct link to it, right? Or can you? I tried, and I didn't.
Yeah, I'll send you something.
All
Maybe you want to put that in the...
Yeah, that would be good. I know, because if people go to the page and they don't find it right away, I know they'll leave.
It's frustrating, yeah.
All right, should we go right to some picks?
Yeah.
Is that good?
I think we probably ought to at this point.
Yeah. All right, I need about 10 minutes to scroll down here. Hang on.
We have a little email accumulating.
Yep,
Dickson, do you have a pick of the week?
I do.
What might it be, sir?
It's a website that analyzes tornado activity throughout the United States. I was prone to pick this because yesterday there was a severe weather warning for Wichita, which is where my son lives, one of my sons, and he's an air traffic controller, so I presume that this will affect his life directly or indirectly, I'm sure directly, by diverting airplanes around these tornadic activities. And it's an interesting map because I spent some time in Indiana as a graduate student at the University of Notre Dame, and I learned then that Indiana is one of the states with a very, very high number of tornado warnings, but very low number of actual tornadoes. Whereas the state of Texas has tons of tornadoes. I mean, lots of them. More than you would think for a state that's, I thought it was below the tornado alley, but apparently it's right in the middle of it. And then, of course, we all think about Kansas and Oklahoma as the hotbed for these things. But I see one tornado happened out in Oregon, another one up in Maine, so they can occur, all over the place. It's quite...
At 5 in New Jersey.
Yeah.
In Union County, that's where I live.
I know, we've had tornadoes.
I never saw them.
They can occur anywhere in the country.
Correct.
No, they can't occur here.
Apparently, they're getting...
Let's see what they have in Michigan. All right.
We had one...
It says 8 in June.
8 in June.
I don't know.
So
where
we're not
you
even
are.
in the
You're
not even in the maximum.
Southeastern Michigan, Kathy. Is that
Right.
Yeah.
What,
Washtenaw County.
Washtenaw. 8. Whoa. Now, what's this Washtenol? What's that? Is that like a native?
It's a Native American word, yeah.
Cool.
Right.
Shiawase.
I found it interesting just to look at the patterns and to see how from year to year the patterns don't vary very much. But of course, I watch the Weather Channel every now and then just to catch up on my geography. And Al Roker has a wonderful delivery system. And they were discussing tornadoes yesterday and showed some of these maps, and that's where I got the
Rich,
idea.
what county are you in?
I'm in Alachua. We got 6 in June.
Another Native
So
American.
according to this, I'll tell you, you don't want to live in Nebraska.
No.
We got Cherry, Nebraska, 42 tornadoes in June.
Wow.
Are you in Berkshire, Alan?
No, I'm in Hampton, which had 9 tornadoes
9,
in
yeah.
June. One of them a few years ago was actually quite bad. It did some significant damage. It happened to touch down right in and near Springfield, which is
Wow.
the city just north of me, and took roofs off a bunch of houses, and I think killed a few people and just damaged a whole bunch of stuff.
Yeah.
Dickson, are you in Bergen County?
You
had 5. You had 5.
I know.
Yeah.
Yeah.
Wow.
Elbert, Colorado, 53 tornadoes in June.
Unbelievable.
I'd moved to Nebraska from Elbert.
That's crazy. That's right.
Well, I visited.
That's right.
I visited Kansas last year, and everywhere you go, you can find tornado shelters. There's a lot in Kansas.
Exactly.
46 in Barton, Kansas.
Yeah. In fact, when you land at the Wichita airport, where my son works, the gift shop sells these little ruby slippers.
Yeah. famous.
That's great.
Yeah, don't forget your ruby slippers.
right.
Lovely.
That's right.
Lovely. Thank you.
And your little
Dickson.
dog, too.
That's right. Toto. That's right. Which, by the way, is I think a Swahili word for child.
Toto?
Toto. I think that means child in Swahili.
Thank you, Dickson.
You're welcome.
Alan, what have you got for us?
I have an RFI that the NIH put out. This is a request for information, and boy, I'm sure they're going to get some. They call it Optimizing Funding Policies and Other Strategies to Improve the Impact and Sustainability of Biomedical Research. If you read it, it basically is, okay, everybody, how do we fix this mess?
Funny.
So they want to know how to make the biomedical research enterprise sustainable in light of the current environment, and they're looking for ideas for policies and such that would get to that point.
Read the Gingrich op-ed piece.
That's easy. Double the budget.
Yeah.
Yeah, but I agree with Richard entirely. I think
Put it all in RO1.
I totally agree with that. Absolutely.
Well, here's the form.
and maybe not.
They're going to be swamped, but the deadline is May 17th.
Should only have a maximum of 2 RO1s per investigator. That's another thing that I'm very passionate about.
Sure.
And people with many more than that.
Fill out the form, Dickson.
There you go.
Rich, what do you have?
This is another pick that originates from my son-in-law, Joey Maines. If you'll recall, I had one a while ago on mutagenic chain reaction that raised the ethical questions about ultimately about engineering human embryos, and he sent me an article from Popular Science titled, "For the First Time, Researchers Edit the Genes of Human Embryos." And this is a Chinese group that published a paper. I found the original paper, and it's published in, let me get it here, Protein and Cell.
Yes.
It's a Springer journal, and it's titled CRISPR-Cas9 Mediated Gene Editing in Human Triponuclear Zygotes. So it's not, depending on, we could discuss this, it's not quite as bad as it might seem from the Popular Science article, where it says, rumors have been buzzing for months that a team of Chinese researchers was intent on editing the genes of a human embryo, and basically talks about how there's been concerns about ethics, but these guys aren't listening. So apparently a common non-viable byproduct of in vitro fertilization is embryos that have 3, or zygotes that have 3 pronuclei. Ordinarily, when you fertilize an embryo, you get 2 pronuclei, the moms and dads. I don't know how you get 3, but I gather they are easily recognizable and would be thrown away because they're non-viable.
Right.
And this group of researchers used those to experiment with whether or not you could actually use the CRISPR-Cas9 system to do editing in a human embryo. And, you can, if ultimately that's the kind of thing that you want to do, you could justify this sort of approach. And they tinkered with the human beta globin gene, which is a reasonable target because it's a common site of genetic disease that you get mutations that creates a disease called thalassemia. So basically an anemia type disease. But interestingly, what they report is that it worked in about half the embryos that they got, but it wasn't necessarily always accurate, and there were off-target effects, and so it's not ready for prime time. So,
I mean, this is true even in the lab. If you're doing research, if you're trying to knock something out in a cell, the original promise was that it would have fewer off-target effects than siRNAs, for example, but it's got problems also. It's not perfect.
Right.
Yeah.
Right.
Unfortunately, yeah.
So it's an interesting question as to whether these guys should be doing this stuff at all. But there will be discussion of that, put it that way.
So there has not been an official moratorium yet, right?
No.
It was just a call for one a couple of weeks ago that we talked about, right?
Right.
However,
and the Popular Science article concludes this paper was rejected by both Nature and Science because of the controversy surrounding it. So I guess next time you have something rejected by Nature and or Science, you could just say it was because of the controversy surrounding your work.
Yeah,
totally.
All right, thank you, Rich. Kathy, what do you have?
Well, as you know, I co-teach an undergraduate virology class, and the last day of class was on Monday. Oh. We have a thing in the class where the students can earn about a little less than 10% of their final grade in a variety of ways. And for the second year, we offered them this option. We had even more students take us up on the creative presentations. And the inspiration for this, for me, was that I had just seen Sheldon Campbell, the singing microbiologist, where he had put words to tunes and accompanied himself on guitar. And we just opened it up to allowing them to do it any way that they wanted. And we had one person come in live and do a whole string of songs on guitar that were really great, a lot of really funny words to those. And then we had a couple do YouTube videos, so I have the links for those. The first one was stop motion animation, which I sent to you guys after the
first.
Yeah, I looked at it. Very good.
Really, really well done on HSV1. And they can choose any virus, one that we have covered in class or one that we haven't, for these creative presentations. And the second one that I'm going to mention, also YouTube, she made a comic book about HSV1. So both of these, I think, will be really good study aids for students for next year's class, and one of them was in time before their final, and so if they'd watched the video and really paid attention to it, it could have helped them with several points on the exam. And then we had people do kind of a half live, half video skit, and one person read 2 poems that she wrote that have really good rhymes and scientific content, and that one, so Vincent has put the link in there to that. So they're kind of rap-like, and so she read it in class, and that was really great. And then another one, a guy did, and we have the Google link for that, about H5N1. And they're just great. We got the students' comments on them too, because Beth Moore and I are both kind of so overwhelmed by these that we can't really be very critical because they're just so creative and we really like them. But the students pretty much agreed. We had them rate them on scientific content and creative genius, 2 separate scores. And it's been really fun. And one of the students came to me specifically and said, this is the first class where I've ever had a chance to do something like this. And it was really worthwhile. So
Great.
We enjoyed it.
What did you tell them? You could do anything creative, or did you give them some guidelines?
We tell them they can do something creative about one of the viruses, and then we have them run it by us about halfway through the semester. We say, okay, tell us what you're doing, so that we know that at least they're not going to leave it to the last minute. And so it's just something about virology. One person did a song about vaccines and vaccination.
Yeah.
So
and it's 10% of their grade, right?
A little bit less than 10%. And then some of them choose to do just straight up PowerPoint presentations. Some of them choose to have an assignment where they can listen to a TWiV from a list that I give them and write about one of the papers that we discuss, and then go on and say what future directions would be, propose a hypothesis. And then some of them, about 10% of them, take a final that we write about all of the presentations that are given in class. So, yeah.
That's cool. I might do that next year. I'm always looking for...
So did any of the YouTube presentations go viral?
One
pun intended.
Has at least 98 hits. And I had sent them to Sheldon Campbell, the singing microbiologist, and when I looked at him just before the show today, he made comments on both of them.
Yeah, that's good.
I sent those to the students.
Very
I said,
nice.
Sheldon Campbell commented on your YouTube, so I think that'll be cool.
That's cool. That's a good idea. I think they're always... I mean, I have 150 kids in my class. I would hope that all of them wouldn't pick to do videos, but they might.
Right.
And they would I give them other extra credit, but this is a lot of fun, and they could do it next year. I think that'd be fun. My pick is, I found a cool article on Ars Technica about the Apollo 13 mission 45 years ago. And it's really nice because it goes into exactly what went wrong, why the oxygen tank exploded, and the sequence of events, which is really interesting. This oxygen tank had been slated to be used on an earlier mission, and it was taken off. It wasn't used and was dropped. It was dropped just 2 inches, but it caused some internal damage that was never picked up. And it goes through a chain of events that occurred and eventually led to it was put on the Apollo 13 mission, and then it blew up a few days after takeoff. But it's really interesting. And what's cool is that there's a link to a longer article which has even more detail. It's in IEEE. And finally, there's a little video which is of mission control at the end of the mission. So what they did is they turned on the lunar excursion module and got their life support from that for the mission. And at the end of the mission, they jettisoned that before re-entry, and one of the mission controllers says, "Farewell, Aquarius, we thank you," which is, it's so cool. But it's really a nice read.
Was that really 45 years ago?
Isn't that amazing?
Holy cow.
You're not even 45, Dickson.
That's right. That's exactly right.
Anyway, it's a cool read. It's a fun read. The short one is very fun, and you should look at the video. It's like 10 seconds with the old scratchy mission control. Farewell, Aquarius. We thank you. And this thing floats away, and of course, eventually it burns up. It's just a machine, but it saved their life. It's cool.
Amazing event. That whole thing was an amazing event.
They also commented in the beginning about the movie, which, of course, was made with Tom Hanks.
Tom Hanks, yeah, sure.
He said, in the movie, they're yelling at each other. And he said, these 3 astronauts never raised their voice. They're as cool as cucumbers. We have a listener pick from Johnny, who sent us a video, the story of Rudolf and Leopold Blaschka. It's called The Botanical Wonders, which is these people were glass makers. And they make glass
flowers
and stuff.
You don't like that, Vincent. You shouldn't have.
I'm not saying I like it. It's a pic of Johnny And this is a movie about the Corning Museum where they have an exhibition for this. And
actually, one of the biggest exhibitions is at Harvard, the Harvard Museum of Natural History.
Yeah,
and I've seen this thing, and it is stunning.
Phenomenal.
It's amazing.
That's true.
Oh, Johnny's right there. She could go.
Yeah.
Anyway, I think she picked it for you, Dickson, not for me.
She did. That's
you
true.
like flowers.
I love flowers.
You're writing a book about flowers.
This is true.
Cool.
Yeah.
Wow.
It's called Duets.
Do I have to like it?
No.
You might
Kathy,
have to
I'll send
read
you a
it,
copy.
Vincent.
I will send you a copy because I know you
know
like
what,
flowers.
Rich? I've already read it.
He helped me make it.
Help him
actually.
put it together.
He helped me work out in design. I'm not as facile with that as Vincent.
I have to put the text and the pictures together.
We did that. We did that.
I mean, flowers are okay.
Yeah, they're fine.
I know that they're needed for the world and all
They're
that.
beautiful.
But they have viruses, as you saw
They
today.
They do.
All right, that's TWiV 334. You can find it at twiv.tv and on iTunes. And please send us your questions and comments to twiv@twiv.tv. You can find Dickson Despommier at this stream in Pennsylvania where he fishes.
That's very true.
And also at Racaniello.org. Thank you, Dickson.
You're welcome, Vincent.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun. And I really liked the plant virus paper. It was a fun read.
It was a fun one, wasn't it?
Mm-hmm.
Yes. And thinking of wine, gee, maybe I'll have a glass tonight. Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
Where there are, 9 tornadoes last year or something?
6.
6.
I didn't see any of them. No big deal.
Thank goodness.
Alan Dove is at turbidplaque.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.